Good afternoon, ladies and gentlemen. It's quite a great pleasure to welcome you here in Bangkok. And I'm pleased, and I will start by the ladies first. I'm pleased to introduce first Dr. Anne Collin from INRA, in charge of the, I would say, conditioning of the young birds and how you can influence the first future growth of the broilers. And Dr. Cécile Berry, director of the Poultry Research Center in France, the BOA new name of the institute, and Dr. Jenny Rura from Queensland University in Australia, and very well known on the appetite control, feed intake control, and so on. And you know, we are organizing a seminar tomorrow focusing on rearing birds under hot conditions. And one of the first aspects when you talk about hot conditions is very often the feed intake and the drop in feed intake under hot conditions. And my first question to all of you could be, do you think that we can change a little bit that and stimulate the feed intake, and that could be applied also under hot conditions?
Well, I guess I will start, and thanks for the invitation. It's a pleasure to be here. The quick answer to the question is yes. I think things can be changed. We can modify. We can stimulate or reduce intake, and even under heat conditions, there are certain parameters that we can work on. Now, the question of can we modify feed intake needs probably to go together with, if we modify feed intake, is that going to have an impact on growth and feed conversion? Because what we're saying is that not always that you have a response on feed intake, you have also the desired response on growth, particularly under heat stress conditions, right? So I think that the answer is complex. So can we change feed intake? Yes. Is that change in feed intake going to be translated into meat, if you want? Not always. So we need to be aware of that, right? And I guess, I don't know if energy is always a question.
Yeah, I think you're right when we say stimulating feed intake. Obviously, the metabolic efficiency under hot conditions is also a parameter. But I would say when we talk about stimulating feed intake, can we also manipulate or stimulate the bird early in life, even in the embryo phase, to stimulate the appetite?
For the moment, I don't know really any study about this, but it could. I think it's a field to explore. I think that beside this aspect on feed intake, the animal has to be also able to dissipate the heat it will produce with this load of heat, additional heat. So I think that by favoring heat loss also, perhaps it's a way to have the animal lowering less is feed intake.
If I may add to the question, because in the area of nutrient sensing, how the bird is sensing nutrients, basically whether it's in the oral cavity or in the GI tract, there's been some recent attempts to modify those the expression of these genes in ovo or perinatal, if you want, and with reasonable success. So you can basically, I think you mentioned before when we were chatting informally, the concept of conditioning, and I think you do. I think you can condition some of these genes, and obviously we have an expert here on epigenetics.
You can comment more on conditioning.
In fact, we have done some experiments with heat conditioning during embryogenesis, and it has a positive impact on thermotolerance when you challenge the birds with heat in the long term. It also has a positive impact in the breast yield. It lowers fat yield at the abdominal level. And in fact, there are other strategies to be studied, perhaps linked to the nutrient load in the egg that could also have an impact on the further metabolism of the bird. And here there are probably many things to experiments also to do with this.
And when you precondition the animal, in fact, what is the mechanism at the end to be adapted to the heat, to the future heat stress? Is that you improve the heat loss, or you change the metabolic production of the heat to be more resistant?
I think we have both types of mechanisms. The animal will reduce its energy metabolism, the intensity of energy metabolism. We have a lower plasma T3 concentration, for example. That is an hormone that is stimulating heat production. So this is decreased. We have the expression of some genes involved in mitochondrial metabolism that is decreased. Also in the liver, decrease in the activity of citrate synthase that is involved in Krebs cycle. But also we think that the heat loss mechanisms are also affected because in the animals that were early conditioned, they are more able to evacuate heat by the comb, we think, with a sensible heat loss. And we also have results on gene expression in the muscle that are induced in case of heat challenge, and they are involved in vascularization. So we think that at the level of blood supplied towards the periphery of the animal, perhaps there is here a mechanism that could adapt better the bird.
Very, very interesting. And I think to jump on that, when I see lower T3, change of the mitochondrial functioning, change in fat, vascularization, what does that mean in muscle development, mid-quality after?
Yes, there is a few data regarding the impact of early management, and especially early conditioning or very early feeding on muscle development. And we know that this can really affect the future development of the muscle. And this is very important, especially now, because you all know that there is many defects related to cellular disturbance, like white stripping, wooden breast. And we know that there is a replacement of muscle fiber by adipose tissue and connective tissue. And this lead to all these defects. And I am convinced that by the early management, either early feeding or change of the in vivo condition, temperature or oxygen supply, for example, it would be very interesting to know how to stimulate the capillaries formation.
And that would be also interfering with hot conditions, for instance? Do you have any idea about the interaction with the environment?
Yes. I think that hot conditions are really inducing oxidative stress. So, and we know that in case of defects, meat defects, the oxidative stress is very important. So, in hot condition, I think the result and the problem will be worse.
Could we even reinforce that, looking at the breeder side before, even before the embryo and the early feeding?
Yes, there are more and more studies showing that some pathways can be affected in the parents. And so the breeders, and then could pass to the offspring. And currently we have some programs to try to unravel whether the thermal conditioning of the parents can affect the yes, the offsprings. And especially my colleague Vincent Coustam, who is an epigeneticist, is working on a quail model that of inbred lines in order to limit the genetic variability, in order to really study the mechanisms in the parents and in the offspring to see if there are epigenetic mechanisms that will affect gene expression and then the mechanisms involved in thermal tolerance.
Very
interesting. And maybe you want to add something?
Yes, I was just going to say, and I think it's a very interesting concept, because and it relates a little bit to what we're doing. We've been trying to identify what nutrients are, or what genes related to nutrient sensing are affected by a situation of heat stress, right? And we're hoping that we're asking the animal to tell us, what's bothering the animal, or what genes that are related to nutrient digestibilities, nutrient absorption. What are those genes that we see being overexpressed? And we have, and I think most of the genes that we have identified, genes of interest. Now, we're not talking about RNA-seq, where you look at all the transcriptomic. Looking at specific genes, and they are related to amino acid sensing and amino acid absorption transporters, and I think that's telling us a few key aspects of what can we then go back and says, all right, can I get this animal ready for the heat stress before the heat stress comes? And how do we do that? Well, if we know which are the genes that will be key, can we then use conditioning or parental feeding, and I think there's robust evidence, at least in nutrient sensing, that we can change that early in life or in novo, that we can expose the embryo to a number of different ligands, if you want, or nutrients, and get that embryo more sensitive, more ready to absorb those nutrients. Who can do that? I think that's a future path that somebody will need to take into account, right?
In this line, we have very recent results at the lab showing that in the hypothalamus of the quails submitted to the heat stimulation during embryogenesis. In fact, there are some pathways affected related to epigenetic modifications, and some of them will regulate pathways linked to neural response, but also gustation. So perhaps there could be here a link. Yes, with this.
We definitely need to talk.
It's good to meet during seminars and so on. Maybe another question. We talk about nutrients. We talk about energy, amino acid, and so on. What about water? Because in fact, under hot conditions, the first thing we drink is water. So how can we stimulate the animal to drink more? We know that when they drink, they eat. So.
Definitely, and when we talk about heat stress, it seems that a lot of the changes in metabolism around heat stress might be one way or the other related to redistribution of liquid, of water in the body, right? We know that the heat stress response is about adrenaline, just getting the blood pressure up in periphery, reducing blood in the GI tract, therefore creating leakings in the gut, therefore creating inflammation and so on. So there's a number of different aspects of how not only birds, I wouldn't say that's unique to birds, but how we deal with water reservoirs in the body to account for heat stress. And obviously one of the things that we will see, at least in humans, is dry mouth, right? And dry mouth goes exactly against any intake at all. So if you have dry mouth, the last thing you wanna do is eating a cookie, right, or a meal, right? You're not going to be able to. Birds, in fact, when it comes to salivation and lubrication of the bolus, are very poor doing that. They have very little saliva, and the saliva barely gets into the bolus itself. So a little effect on that hyposalivation, and you have a problem. The bird would not be able to swallow. So I'm quite sure that water is a major player in heat stress. I'm not sure we're dealing with water appropriately. I'm not sure we do. But certainly is. And if you want to get that spin on how do we sense water, well, we know how we sense water. We know the receptors, aquaporins are called. They are present in the taste buds, and they are present in obviously ubiquitous. It's a big family, the aquaporins, with a number of different functions. But obviously one of the functions of aquaporins in the oral cavity is perception of water, right? And we know that aquaporins will be upregulated when there's some dryness, and so there's craving for water, right? So,
have you ever considered the water regulation?
Not in mine, but there are studies about electrolytes and salt, potassium chloride into the water. I think the quality of water also is important to manage. But we have to take care of the litter also when we use such strategies, I think.
And regarding the meat aspects and so on, have you even looked more at the effect of the hot temperature on the quality of the meat, on the response of the muscle to the hot exposure and so on?
Yes, of course, hot temperature will affect the development of muscle, and by this way it will affect meat quality, and because it will affect the growth. So, for example, if it will have an impact on the incidence of defects, because we know that white stripping, wooden breast, spaghetti muscle are more frequent when the growth of the muscle is very high. So in some term, the hot can preserve
could reduce
and reduce the incidence. But we have to be careful, because we know also that oxidative stress is one of the problem in this defect. So hot condition can have an impact. And I would like also to say that the response is not so easy, because it depends if the stress is chronic or if it's acute stress. And it will also affect the glycogen reserve of the muscle, for example. And the response are not always easy to interpret, but we know that it can affect, for example, drip loss, and have a lot of consequence on processing yield and on production cost. So it's important to study the effect of hot condition on meat quality, and I think we have some work to do.
Maybe Jenny, you want to compliment?
Am I allowed to ask a question? Because that's
no problem.
It's fascinating. I'm really interested in meat quality as well. And what I would connecting some of what we've been looking at, the different pathways that suffer a change due to heat stress, and one is definitely the non-esterified fatty acids, the NEFA. So the whole fatty acid digestion, absorption, mobilization, it's all over the place during heat stress. And I know that obviously chicken meat doesn't have a lot of fat, but still I think it could potentially, at least to my limited knowledge, tremendously affect the taste, the texture, the succulence, if you want. Would that be reasonable to assume, or do you know what happens with fat and fat deposition during heat stress?
I don't know if I can answer. The thing is that the breast muscle is very poor in lipid, and we know that it's quite difficult to change it. Actually, the best way to change it is to have a white stripping, and we know by survey in slaughter plant that with years the amount of lipid content increase from 1% to 2 in I would say 10 years about. So that's but in the same way it has a bad impact on quality. So I think the succulence is not really related to this change. What I would like just to add about the meat quality, I think it's very important to manage the early development of muscle, because we know that during the last days during incubation, the yield regarding body weight of the breast muscle decrease because of protein catabolism, because at this time the chick, the embryo needs a lot of energy. And we know that in the modern genotype, they have really a lack of energy reserve in muscle when they hatch. And this, we demonstrate very recently that this lack of energy very early can impact the meat quality and can impact the incidence, for example, of white stripping. So I really think that people who work on meat quality has also to work on this very early development to try to understand if the modern genotype lacks something at the very beginning, some nutrients, some oxygen, and
so finally, when we talk about all these, I would say, myopathies or breast defaults and so on that are developing, I would say, in temperate conditions. In fact, even under hot conditions, you put another stress on the top of the animal, so you might also find it. And I've heard earlier on today that people are now starting to complain also here about these defaults. So I think really it's something that will spread, and that is important to consider. Obviously, the early management, the breeding management, the early conditioning is maybe an issue, but I think there are still, sure, many things to do. So I appreciate our discussion this afternoon, and I think we could last for hours on the topic. I'm sure that you are all passionate by your topics, so thanks very much, and hope to have enjoyable talk tomorrow.